Okay.
Coding in progress. I
Okay.
think probably Thomas, this is yours. I'm still keeping it on making coffee.
Okay. Good morning, everyone. It's my pleasure to introduce today's distinguished colloquium speaker. It's very weird. There is a time delay. Okay, so uh, Dr. Hillen is a world-renowned mathematician specializing in mathematical biology with an impressive academic career that bridges mathematics and its application to the life sciences. Professor Hillen is a faculty member in Department of Mathematical and Statistical Science at University of Alberta, where he has been a leader in advancing research areas such as population dynamics, cancer modeling, pattern formation. And uh, Dr. Hillen is also well known for his textbook, Mathematical Modeling with the Differential Equation. Okay, so today he will talk about modeling microtube-driven invasion of glioma. So let's welcome Professor Hillen.
Yeah, thank you very much, Jay. So it sounded like you were waiting for your own echo in the room to die away. I hope my voice is okay. If my voice is not clear, just let me know and I can make
Perfect.
changes. It's good? Okay. Yeah. So it's a great, also a little bit surprise to speak at a little conference. And so I assume that the people in the room have made very, very mixed background. So maybe some are experts in PDEs and some are experts in things. I have to go maybe slow enough so that most of you can understand everything. So the story is about glioblastoma. Glioblastoma is the most severe brain cancer. And upon diagnosis, people often have a survival up to 12 months or 12 to 15 months, even with treatment. So glioblastoma, or in short, glioma, is the most severe brain cancer there is. And so the modeling here, or actually it's a story. So the story I tell you is about the modeling of glioma. And yeah, so the work is based on these three papers. And I show this to show my co-authors, so Nadia Loy, Kevin Painter, Ryan Thiessen. This is about the modeling, which I will talk about. It's in the Journal of Math Biology. Alexandra Shyntar is a graduate student of mine. And then Ryan Thiessen is also a graduate student of mine. And then they have worked on other aspects. And it depends a little bit on the time I have, if I can talk about their papers as well. We have to see. So let me show you pictures of glioma right away. So brain cancer. These are brain MRI scans. And what you see these light regions, right? So this whitish region here or there or here, which is the cancer. But then if you try to find a boundary of the cancer, this will be very difficult, right? So even if I take a pen and then maybe I kind of delineate here the boundary of the cancer, I might get something like this, but it's not very clear, right? So the invasion of the cancer is very diffuse. And this is even more so here. So maybe I put this, so it's very diffuse invasion, very difficult to say where the tumor ends and where the healthy tissue starts. And this is a problem, right? If you want to treat cancer, maybe by radiation, you need to know exactly where you radiate and exactly where you don't radiate. You don't want to damage healthy tissue. Okay, so it's a very diffusive cancer. And then the first idea which people had to model this is a diffusion equation, right? If it looks so diffuse, then maybe a diffusion equation or a reaction diffusion equation might be the best way to model this. And this has been done. So now I show you the three most prominent diffusion type models for the glioma. The first one is a model by Christine Swanson, which she developed in 2000. So it's already quite several years old. It's called the proliferation infiltration model, or the PI model. And let me just write it down for you. It is just du by dt equals d of x d square u by dx square plus mu u 1 minus u divided by k. So this should be an equation that at least people who took a PDE course with John Jung Kim know really well, which is the Fisher KPP equation, right? This is the Fisher KPP equation. So what does it mean? So we have a population density of cancer cells which depends on space and time. And then we have a time derivative, so which is the change of time. This here is a diffusion term, second derivative in space. And then this is the logistic growth term. So it's a little bit, I'm a little bit guessing what the audience knows or doesn't know. So maybe I'm a little bit too slow, but it's probably fine. So that's the logistic growth, and so it's essentially really the first idea which you would come up with if you try to model diffusive invasion of something which grows. And then the diffusion coefficient here, Christine uses two values, 1 for gray matter and 5 for white matter, just indicating that glioma grow faster or spread faster in white matter. And white matter and gray matter are the two main constituents of the brain. So gray matter is where the thinking happens, where the brain function happens. And white matter is the nerves, the nerve connections, so the connecting fibers of different regions in the brain. So this is the proliferation infiltration model. The point I want to make is that it is based on a very standard kind of diffusion term. Okay, the second class is go or grow models. They started to become important around 2012. There are several papers in the same year who studied the go or grow mechanism. And here the idea is you split the cells into two parts. You have an invading component, which is called grow component, a go, and you have a growing compartment, which does proliferation. So meaning you have two populations, one for go and the other for grow, and then we have these equations here. Let me do this just quickly. This is the diffusing compartment, d square u dx square, and then we have dv dt equals f of v, okay? And then they are coupled. So we have like minus alpha u, and they go here, plus alpha u, and then minus beta v, and they go here, plus beta v. So we have these two processes of diffusion and growth, but they are now coupled in different equations, and they're switching between those with these alpha and beta terms like this. And so these are called go or grow models, and they have also been used a lot for glioma modeling. A little bit more complicated because they include this go or grow switching. Okay. And then the third class is the anisotropic diffusion models, and this is something I have worked on myself with Painter and Swan and Zagorlescu and Engber and other people. And here the key is that we use anisotropic diffusion in direction of white matter fibers. And now this term looks really funny. So this is still the diffusion term, but now I have a diffusion tensor or a diffusion matrix. And then the logistic growth I call f of u. Okay, and so this is called anisotropic diffusion. And of course, I need to explain what this is. So the experts among you will understand this very clearly. But so the diffusion term d of x is a matrix. Okay, so it has entries dij, which can depend on x, ij. So you should really imagine an n by n matrix. And then this colon product here, d of x u. So this is a full second order derivative. Ij is 1 to n, d by dxi, d by dxj of dij of x times u. Okay, so it's a full, the sum of all possible second order derivative combinations you can make using the matrix dij. And then this Dij here is informed from diffusion tensor imaging. Which means we have medical data which we can use to estimate this Dij. And once we estimate the Dij, we can put it in the above equation, and then we have a good model for glioma spread. And if you stay awake until the end of my talk, then you can see how I apply this to patient data. Okay, so where is my 3 models? Here. So we have the simple Fisher KTP, the PI model, we have the Goa-Grow model, we have the anisotropic model, and they're all based on the idea of diffusion, which is the idea from these images that the spread of the cancer is diffuse. However, as we did this, like in 2012, 2013, nobody knew how glioma cells really spread. Nobody knew how they move into the healthy tissue. And this is a problem because, of course, we want to know how they move. Until in 2015, Oswald and collaborators published experiments on mice where they have a living mouse, and in the living mouse, they can visualize the growing tumor in the brain. This is completely crazy. So what I'm going to show you actually right here is one of these images on a living mouse. Okay, and so let me talk you through this first, and then I'll show you a video in a second. So this is the measurements by Oswald. And what you see in green is the cancer cells, glioma. And red is the blood vessels. And everything black is healthy tissue. So you see how the cancer cells invade into healthy tissue, which is where there's lots of blood vessels going on. And the cancer cells really are these bean-shaped things. So these are the cells. And they send out very thin extensions, very thin protrusions. So maybe here. And these are called microtubes. Or we call them glioma microtubes. So this cell here sends out one of these microtubes, and then they grow into the healthy tissue and they test the environment, and they can make connections to other objects. And what then happens is, upon mitosis, you get 2 nuclei, but the cell does not divide. You only get 2 nuclei, and one nucleus moves down the microtube and maybe settles somewhere here along the microtube. And then at this location, you can get a new cell, and then this new cell sends out new microtubes to invade further into the healthy tissue. So it's a process of microtube-driven invasion where nuclei move along the microtubes and then develop into cancer cells or bulk cancer cells. Okay, so that's my description. So are you ready to see the videos? I think you are. So let's see if this works. Come on, open up. It worked earlier. Maybe I have to yeah, so here we go. I start again. So this will be a little bit delayed, but I hope you can see. So you see from day to day how from top to bottom the tumor invades, and it uses exactly the process I described. And then in the second part of the video, you see individual microtubes, and I'll stop it for a second. Okay. In the box, you see 2 microtubes. They're actually 2, they're sitting behind each other. This is not a splitting. And you will see one end extends and the other end retracts. So one goes forward and one goes backward. And I start it again. Maybe you can see this. Yeah, and so this was super quick. I run this entire thing again. Let's run it once more. So first of all, you see the cancer invading from top to bottom using this microtube method. Okay, and then in the second part of the video, you see individual microtubes moving. And they are really, really long, right? You see the cell is far away. And so, yeah, so that's the process. And then they form a network. They form a network of cancer. And then within the network, they can release signaling. And this is the other video I like to show you. This is this one, where you can see calcium signaling inside the tumor network. So the green cells here are tumor cells, okay? And sometimes you see flaring up, which is signaling with calcium waves. And this is entirely crazy. I mean, imagine, I mean, this looks like a brain, right? This looks like a functioning brain. But in fact, it is a kind of a functioning tumor. So you could say the tumor tries to think. It tries to communicate, release action potentials, and build a network of communication. So completely crazy, at least from my point of view. So let's see until the thing comes back. So there it is. Okay. So then what we saw is a microtube-driven invasion of glioma, and we did not see a diffusion equation, right? Looking at these details, it didn't look like a diffusion process. So the question is, are all these models then wrong? So all these models here, are these actually wrong? They do not seem to be supported by these videos I have just shown you. Okay, so that is the question.